Welcome to the lecture series on vitamins and nutrition. Today we are going to continue from the previous session on the mechanism of vitamin K, especially its role in clotting pathway. So today we are going to look at the aspect of how vitamin K is basically getting activated. In the previous session we saw how the quinol form is extremely essential in its active version of vitamin K to initiate the reaction, and I want you to recollect this particular terminology, what we call it like gamma carboxylation. So gamma carboxylation is extremely essential because that imparts the carboxyl group to vitamin K. Now the carboxyl group is basically added and vitamin K acts like a kind of, I would say, the cofactor to carry out this particular reaction. So addition of carboxyl group is that particular part which actually provides the chelating ability for prothrombin. So we saw exactly how this is carried out, and we saw the, I would say, the cytoplasmic pool of vitamin K, and now vitamin K is being constantly replenished to carry out this particular reaction. Okay, so now today we are going to look at some more aspects of how this particular mechanism happens, and we are going to look into the interesting aspect of how we can look into other related proteins where vitamin K plays an essential role. So we saw all this, and we saw how different enzymes are inhibited warfarin, which is an antagonist for vitamin K. Today we are going to look at how it's basically the application of vitamin K can be extended to other category of proteins. So we saw in the previous slide, this is the pathway. Actually we were looking at how you add this carboxyl derivative over there as part of gamma carboxylation reaction, and here you see how the glutamic acid proteins are getting converted to the GLA proteins. Okay, you see there, this is a very concise mechanism. So vitamin KH2 getting converted to vitamin K oxide, and this is again back converted to vitamin K, and warfarin blocks this particular step, and this KH2 how in the presence of carboxylase would convert glutamic acid to gamma carboxyglutamic acid. So vitamin K dependent target protein. So this picture is basically the kind of summary of how vitamin K is being utilized. So you see over there, warfarin can inhibit this step. Okay, so in this particular step the quinol formation from the ketone is blocked, so this enzyme reductase is inhibited, and again in this step the epoxide, it's getting converted again by a reduction reaction, so that you want to, I would say, restore back, remove the oxygen. So any kind of removal of oxygen is also called reduction. So that way in this case addition of oxygen, hydrogen is also called reduction. So this kind of a reductive step is basically removal of oxygen, and this kind of a reductive step is basically addition of hydrogen. So you should recollect the school book definition of oxidation and reduction. So in that way either side, here reduction, here again reduction, and both can be inhibited by warfarin by inhibiting the corresponding enzymes involved in the pathway. Now this is what happens as a cofactor to the carboxylase that generates gamma carboxyglutamic acid. You have this cycle being regularly utilized. So you see there is this key enzymes, two kinds of reductases acting on either side, playing a major role. Now we look at this, how the calcium binding surfaces, basically this is extremely essential. See, if you look into the clotting mechanism, if any kind of calcium, external calcium scavenging compound is added, I would call it like you add EDTA or you add any other compound which would basically scavenge the calcium, can be an excellent anticoagulant. Especially this method is used in the preparation and the storage of blood. So when we want to collect blood from patients for the purpose of storage, or maybe from the donors from the purpose of storage, we add an anticoagulant, and the anticoagulant what we add over there is either an EDTA or a citrate salt. So this EDTA and citrate salts, they actually scavenge the calcium. So if calcium is not there, clotting mechanism is inhibited. Now vitamin K basically inherently, I would say, provides the ability for prothrombin and clotting proteins to hold calcium by carboxylating them and making them efficient calcium chelating proteins. So the moment this facilitates the calcium bridges, okay, so they create a kind of an anchor on the surface of the blood membrane, and in this case we are looking at the blood platelets and endothelial and vascular surfaces between GLA residues to form internal GLA-GLA linkages. In the sense, you have now a very strong glutamide linkages being happened. So you see the calcium acts like a kind of a hook, and not only that, so that it anchors to the membrane, and now you are like bringing up all the membrane together, and now you stick them. So this is very interesting. So the calcium would allow the hook, the different membranes to come closer, and as they come closer, you in the next stage seal them with a covalent linkage. So this kind of a sequential reaction is what exactly happens during the clotting pathway. So this clotting pathway history is quite, I would say, elaborate, although right now in this century we study them everything in place, one step over the other, everything happening in a very sequential manner. But if you look into the discovery of clotting proteins, and vitamin K role had played a tremendous contribution in understanding the pathway, and not only understanding, the information on vitamin K's role in the clotting pathway has actually resulted in many therapeutic interventions. So we have a better understanding of how anticoagulants work, and if you really want to evaluate different anticoagulants based on their mechanism of action, we are able to do it. Everything now has been shed light by vitamin K related or vitamin K dependent gamma carboxylation reactions. So that's why if you have to study clotting mechanism, you have to study about vitamin K's role and its metabolic intervention in making the complement, I would say, succinct or more productive. Not only that, you are going to shortly see how vitamin K dependent proteins in the immune and inflammatory pathway are so well, I would say, focused or put in order so that the pathway can go seamless. So you see there the enzyme required vitamin K, which is basically an hydroquinone and molecular oxygen. So this part we have already seen. Now under low carbon dioxide levels or in the presence of peptidyl, I would say, glutamic acid, see there under low carbon dioxide levels or in the absence of peptidyl glutamic acid, the epoxidation of the vitamin, what we call it like the vitamin K epoxide, proceeds without concomitant carboxylation. So this is a key. So it means what? You can even consider vitamin K directly getting converted to epoxide without actually participating in the, I would say, reaction. So you need to have carbon dioxide. So when you have carbon dioxide, then carboxylation is essential. See, in any kind of pathway, you have many proteins playing their own role. If you could remove one, you can still go ahead, I would say, but it may not be productive. So I can give an analogy. Suppose if a lesson is taught in the class and the hours run and the students are there seated and the teacher is sharing his knowledge, and if the student doesn't participate, the time will go, but the actual delivery of knowledge or transaction doesn't happen because there was no active participation. So same is the case over here. So the vitamin K actually gets converted into epoxide and the reaction is done, but the culprit being low carbon dioxide level. So when low carbon dioxide level, although in a sequential manner it keeps happening like in a cyclic format, the efficiency of carboxylation for gamma carboxylation doesn't happen because now in this case we lack carbon dioxide substrate. Now as I've told you, you have dozens of post translational modification being catalyzed by vitamin K. So the one which we have seen, gamma carboxyglutamic acid residues, is a classic one, and a similar role and related role are being played by vitamin K in variety of such post-translational modification proteins. Now if you look at the reactions, it's quite interesting. So you see around 8 vitamin K dependent proteins in plasma have homologous amino acid sequence in the first 40 positions. See, this is quite interesting to note. So you have almost like 13 clotting factors discovered so far, and their roles have been elucidated. And if you really historically look back, there's been a wide gap of so many years involved, research by independent groups or in collaboration to really establish the flow. Now we have all the information. So you see there, the first 40 amino acids have homology. So what do you mean by homology? So if you take a protein sequence and you align them one over the other, you find how much they are similar in the sequences. You call them like homology, how homologous they are. So they say like 2A, Roman letter 2A, 7, I would say 9, 10, C, M, S and Z. All these proteins exactly are, I would say, homologous in the first 40 residues. And this region requires calcium activity. So you see there, that's very interesting. So if these 40 positions require gamma carboxylation, then immediately as a consequence all these 8 proteins would also need vitamin K. So that's why we categorize them. So you see, that's quite a number. In the 13, almost 8 need gamma carboxylation to activate them, and vitamin K plays that role. So now you see how much vitamin K is involved in the clotting mechanism. To a count of 13, 8 to 13 is quite a good number, I would say. I mean, good contribution by vitamin K dependent functions. So all require calcium, and protein C inhibits coagulation and stimulated by protein S, it promotes fibrinolysis. So whenever we talk about clotting mechanism, it's all very complete, because in one angle clotting proceeds to linkage formation and fibrin clots, but too much of fibrin clot can hamper, I would say, the flow of blood to that particular site. So our system has the other mechanism to balance this. Suppose if there is a clot, fibrin being formed, and you have to dissolve this over a period of time, so that this would encourage the healing process. Yeah, a severe inflammation has resulted, so that all remedial measures are done to immediately arrest getting the situation to a worse situation. You do all the activities, but once the crisis is done, you also have to have a provision to get back to the homeostatic level of maintaining a normal flow. So in that case, the clots which were once formed should not now become an hindrance for the process of healing. So that's what we mean by fibrinolysis. So fibrinolysis is the process by which you crack down all these clots and ensure a normal flow again sets in. So protein C inhibits coagulation, okay, and stimulated by protein S, it promotes fibrinolysis. So you see, the same protein has provision to become like a kind of a coagulant, and also as a provision to break down the clots that resulted from its contribution. So you see the 4 classic clotting factors, 2, 7, 9 and 10 are components of a complex system of protein that function to prevent hemorrhage and lead to thrombus formation. So the protein components of this system, okay, are all called the inactive version, or in other words they are called zymogen. So zymogen is any protease kind of an enzyme in its inactive form. Now you have to convert the zymogen into an active form. Like you could always recollect this example of, I would say, the trypsinogen getting converted to trypsin. So if you ask this question, which is the active form? Trypsin obviously, and let's say chymotrypsin, but which is the precursor form? Trypsinogen, we say chymotrypsinogen. So that's why this zymogen means the inactive version of the enzymes then getting converted to the active form. So you may wonder, Why we study about this? Because most of the clotting proteins are kind of a zymogen which gets converted into the active form. And now if zymogens have to be converted into active form, you need vitamin K. And also remember, why we have to have the zymogens, inactive versions? Why can't we directly have this active versions in circulation and immediately proceed with the action? There is a reason behind it. If you look back, most of the zymogens, and most of these zymogens are proteases. So if you could let these zymogens in the active form, what will happen? They'll start chewing down their own cells and tissues, and it's going to be completely unproductive. Something like when you have weapons, especially highly dangerous weapons, you have provisions to lock the trigger. So something like that. So zymogen is a trigger locked weapon, a protease. The moment you remove the trigger, immediately it starts firing, because there are chances that an accidental pressing of the trigger can lead to violent, I would say, explosion. So to avoid that, we purposely create a lock, and only with full confirmation the trigger can be opened, and only when needed. So zymogen related provisions, and you make them accountable with the vitamin K dependent function, makes it a kind of a flow, so that when it is needed it gets activated, if it's not needed it's not activated. So you see there, that's how it works. So as I've told you, you have to remember this key word, serine protease. Okay, so zymogens are inactive precursors, each of which is a serine protease. So you get the key word when it's a protease. And what is serine protease? In the active site you have serine amino acid, which is an hydroxy group. So hydroxyl derivatives, aliphatic derivatives are there in the active site, and they play an efficient role in the mechanism. So fibrin cross links with platelets to form the blood clot. So you see how it's all linked with each other. So 4 of these clotting factors, 2, 7, 9 and 10. Okay, so they all are zymogens. Now conversion, as I've told you, involves the cascade starting from calcium. So once calcium gets bound with the active protein, it substrate and another protein factor with the phospholipid surface. So it's something like you create the surface to make it little sticky, so that they can, glue with each other. I would say the molecular glue, or in other words the covalent interactions happening between the side chains of amino acids. So you see there how this clotting protein mechanism happens. So any injury would trigger, I would say, a release of lot of cytokines, and most of these cytokines are flagged signals to alert immune system and inflammatory mechanisms to set in, so that they would want to hamper any kind of progression that can lead or go out of control, and that can be deleterious in long run. So we want to avoid all that. Okay, so we say that these signals are amplified. So and moreover, there is a provision for a contained amplification. So it starts little, and as it proceeds, and when the need becomes more, there is a self amplification process. Okay, so the key step is the activation of factor 10, Stuart factor as we call it, by proteolytic removal of the short polypeptide from a zymogen. So you see there the Stuart factor, the 10th factor, is basically the key trigger. So we look over here. So the actions of factor 9, which is basically the Christmas factor. Yeah, you do have names for each of these clotting factors. Okay, so what we consider, which is activated by plasma thromboplastin as a result of contact with the foreign surface. So this is what we call it like intrinsic clotting system. Okay, so intrinsic clotting system starts with Christmas factor, factor 9, and then you have the plasma thromboplastin, and then this is in touch with the foreign surface, okay, creating the kind of a mesh. By the action of factor 7, which is activated by tissue thromboplastin released as a result of injury, what we call it like the extrinsic clotting mechanism. So what do you mean by that? So you should consider clotting is a physiologic mechanism. Generally we are taught in the lower class, whenever there is a cut, yeah, there is bleeding and immediately clotting sets in. That's true. So in other words, the emphasis had been more on the extrinsic clotting system. When there is an injury cut, you try to solve it, but clotting mechanism is part of physiology. It means what? Regularly every day inside a tissue there is this cellular damage and tissue damage happening, and it needs to be sealed, healed to carry over the process. So that's what we mean by how we are going to address this internal inflammatory damages and kind of breaches in the capillaries, how we are going to like really fix them, and that's taken care by the intrinsic clotting system. And in the extrinsic clotting system, it's more of when there is a outward injury happening, how a body responds to it, like a cut, a bruise or whatever. Maybe internal cut, internal damage are all being dealt by intrinsic clotting mechanism. It's not like two different mechanisms, it's the same pathway, and only thing which triggers the event, and based on that you give emphasis. So that's what we say factor 9, Christmas factor, actually proceeds for the intrinsic clotting mechanism, and factor 7 proceeds to the extrinsic clotting mechanism, and finally they all converge. That's always there. So once factor 10, binding calcium and phospholipid, catalyzes the activation of several coagulation factors. Okay, so that's how it works. So the prothrombin, which is factor 2, to its active form thrombin, okay, which catalyzes the proteolytic change in the fibrinogen that renders it insoluble as fibrin for clot formation. So you see that the factor 2 to its active form thrombin gets catalyzed by the proteolytic cleavage so that the fibrinogen can get converted to fibrin. In other words, this pathway might be familiar for you, fibrinogen getting converted to fibrin, and this is catalyzed by prothrombin, which is factor 2. So now factor 5 to its active form factor 5a, that's the second step, and factor 8 to its active form, what we call it, factor 8a. So 5 to 5a, 8 to 8a, and factor 2 becomes activated to give you factor 2a. So you add this a as a subscript to tell you that this particular protein, which was originally a zymogen, now getting activated. So that activated is being written as a subscript called 2a, 5a, 8a. Now this is activated by thrombin in the presence of endothelial cell protein and thrombomodulin. Yeah, you do have other proteins as well. Now we are enumerating all the key proteins involved in the pathway, and their role is almost, I would say, very focused on what they are really acting upon. Now you see it acts in complex with protein S by partially hydrolyzing the activated factors 5 and 8, thus inactivating them. So you see there protein S partially hydrolyzing the activated factors. So means what? When you have an activated factor and they are doing the job of cascading, and suppose if you want to nullify the pathway, you have to now remove the activators. So this activated factors, factor I would say 5 and 8, being inactivated by one more protein. Yes, so activated protein C regulates the activities of, I would say, coagulation factors 8 and 5a, 8a and 5a, and this serves as a cofactor for the activation of factor 10. So I would say the prothrombin mediated cleavage, so it's like linked with each other, and that's the role of protein C. So protein C can be considered as a kind of anti-inflammatory mechanism. So because if inflammation is going to go out of control, you need to bring them back in order. So protein C would be that internal compensating protein trying to fix everything in order. So what we call it like anti-apoptotic activities, binding to the endothelial receptor and cleavage of a protease activated receptors. So these effects involve activation of protease affected receptors, what we call it like PAR1, and the inhibition of NF-kappa B. Yeah, this is amazing. This is the kind of pathway which is directly linked with immune related mechanisms, NF-kappa B. nuclear factor, what we call it like kappa B of B cells. So you see there PAR1 is protease activated receptors. So you see how C plays a role, because C is associated with vitamin K. We study about this. So patients with inherited deficiency of factor C are at high risk of thrombosis. Why? Because the thrombosis pathway initiated through vitamin K will proceed, and there is no way an internal regulator to stop its progress, and protein C would do the job. Protein C is the internal mediator in stopping these mechanisms, because if protein C is not there to like modulate, what will happen? It will throw into thrombotic state. This is something like you drive a vehicle, you have an accelerator and a brake. So the more the throttle, you have more speed coming up, but you do have brakes to bring your speed under control or regulate the movement. So protein C acts like a kind of an internal balancing regulator, bringing the clotting pathway in its proper perspective so that it doesn't go out of hand, because this is initiated by vitamin K, and vitamin K also has this close association. So that's the point we have been trying to emphasize, how vitamin K is so closely associated with all these functions. So now we are going to look at how this mechanism proceeds further. So we have seen how the activation happens and how protein C plays an effective role. Now we have one more protein, what we call it like protein S, which is not a serine protease, zymogen. It's found in the plasma in the free state, okay, and with the regulator component C4b binding protein of the complement system. So complement system is one more pathway. We are not going to deal with that, it's out of scope, but I would say complement pathway is part of the innate immune responses in creating membrane attack complexes to leak the cells, especially bacterial cells. They are not like made for our own body cells, but mostly made against those cells which are invading our cells. Okay, so here we find protein S plays a regulatory role in complement mechanism. So whenever a protein is going to play a major role in one pathway, doesn't mean that it should not play any role in the other pathway. So protein S is basically not a serine protease zymogen, but they do have their own way of interacting and strengthening the complement mechanism. So more it has to do with complement mediated pathways. So you see patients with inherited protein S deficiency have recurrent thrombosis. So you see again protein S is a kind of a hampering, and if that role is removed, there would be spontaneous thrombosis occurring. So you see one more protein Z, which is also not a serine protease, which is closely associated with vitamin K, which is a 64 kilodalton, and this inhibits factor 10A, okay, by a inhibitor. So again this is a deficiency which is inherited, has been associated with bleeding tendency in patients with factor 5, I would say, mutation. So you call it like there could be a mutation that could render the corresponding, I would say, DNA to propagate and give rise to many shortage of these proteins, and thereby totally affecting the pathway of inflammation. Now you see there what happens, how vitamin K plays a role. So vitamin K is essential in the osteocalcin, then matrix GLA protein for strengthening the surfaces, and for protein S, which actually has a inhibitory role to play. So we are almost done, and I think we need to conclude. So I would say how vitamin K is playing a major role in the clotting mechanism. So we saw elaborately how vitamin K contributes in every individual process, and how vitamin K helps in the entire cascade of clotting mechanism, and what all different proteins which are closely associated with vitamin K. So in the future session we are going to look directly into the application of vitamin K and its role based on the mechanism whatever we have seen, and also some of its deficiencies. Thank you for your attention.